So in today's lecture, we are going to see limit test for arsenic. Same as that of limit test for iron. In case of limit test for arsenic, we are going to compare the intensity of the color, that to yellow color, produced by the arsine gas with that of the ferric chloride filter paper. आयन मध्ये आपण कसं इंटेन्सिटी ऑफ पर्पल किंवा पिंक कलरेशन चेक केलं होतं of the test with that of the standard one. Same in case of limit test for arsenic, we are going to compare the color sample with that of the standard one. So formation of color in case of limit test for arsenic is the yellow stain. So in case of limit test for arsenic, we are going to convert the impurity arsenic into an arsine gas. And this arsine gas will then react with mercury chloride filter paper and yellow color will be get occur. So for performing the limit test for arsenic, we are going to use an special apparatus which is known as Gudget apparatus. Therefore, arsenic limit test is also known as Gudget test. We have seen limit test for chloride, sulfate and iron. All these three tests we have performed it into an Nessler cylinder, which is a transparent glass tube having a capacity of 50 ml. But for arsenic, we are going to perform the test into a special apparatus which is known as Gudget. Therefore, it is also known as Gudget test. So arsenic, it is a well-known undesirable impurity which is likely to be present into an inorganic pharmaceutical drug substance, and it is an harmful impurity which is present. So all the pharmacopoeias prescribe a limit test for it. What is the main aim of limit test? Firstly, identifying the impurity, and nextly controlling its amount. Since the nature of arsenic, it is very harmful one, we have to control its amount by performing the limit test. And the pharmacopoeial method is based on the Gudget test. That is, we are going to perform it into a specific apparatus. All the special reagents used in limit test for arsenic, they are marked and distinguished by a letter, that is AST. म्हणजे जे काही आपण reagents use करणार आहेत for performing the limit test for arsenic, they are labeled as AST. So what does AST means? It means that they are all should be arsenic free. म्हणजे reagent मध्ये arsenic नसायला पाहिजे and should themselves conform to the test for arsenic. The reagent मध्ये impurity असेल, say for example potassium iodide मध्ये असेल, हायड्रोक्लोरिक ऍसिड मध्ये असेल किंवा नेसेंट स्टॅनस क्लोराइड मध्ये असेल, म्हणजे रिएजंट मध्ये जर arsenic impurity असेल, so we are not going to distinguish the impurity which is present into the medicinal substance, that is inorganic one, and the impurity which is present into the reagent. So all the reagent used for performing the arsenic limit test, it should be free from arsenic impurity in order to check the specific impurity present into an given unknown inorganic medicinal substance. So all the reagents used for performing the limit test for arsenic, they should be marked as AST. That is the reagents, it should be free from arsenic impurity. म्हणजे reagent मध्ये किंवा जे काय आपण samples use करणार आहे, that should be devoid of the arsenic in order to check the presence of arsenic into test. Since we are going to compare the presence of impurity in the test with that of the standard one. So this is the introductory part of limit test for arsenic. Arsenic, it is an harmful undesirable substance which is likely to be present into an given unknown medicinal substance. So it should be devoid of it. Therefore, pharmacopoeia has prescribed a specific apparatus for performing its limit test, which is known as Gudget test. And the reagent used for its performing the limit test, it should be devoid of arsenic. That is, it is labeled as AST. Next is the principle. So the limit test for arsenic is based on reaction of arsenic gas. In case of limit test for arsenic, we are going to convert the impurity arsenic into a gas, that is arsenic gas. Reaction of arsenic gas with hydrogen ion to form yellow stain. जे काय arsenic gas किंवा arsine gas तयार होणार आहे, that will react with the hydrogen entity of the mercury chloride paper in presence of reducing agent. So extra reagent used in case of limit test for arsenic is the potassium iodide. So it is called as Gudge's test and requires a special apparatus. So this is the basic principle. What is the basic principle? That is converting the arsenic impurity into arsine gas. This arsine gas reacts with the hydrogen entity of mercury chloride paper. So this mercury chloride paper, it is placed in between the rubber bunks. So when arsine gas reacts with the hydrogen entity of the mercury chloride paper, this mercury chloride paper, there is formation of yellow stain on this paper. We are going to compare the intensity produced by the test sample yellow coloration with that of the standard one. So this is the basic principle of the Gudge's test or limit test for arsenic. So ingredients or reagent used for limit test for arsenic, it is the potassium iodide, hydrochloric acid and nascent hydrogen. Each and every reagent is having a specific use. We'll study one by one. So basic principle clear hai. We are going to convert the arsenic impurity into arsine gas. This arsine gas will then react with the hydrogen ion of the mercury chloride paper and it will give yellow stain. We are going to compare the intensity of yellow stain produced by the test with that of the standard one. सगळे जे काही लिमिट टेस्ट आहेत, सगळे लिमिट टेस्ट we are going to perform it by comparison method. Either we are going to compare the turbidity formed or either we are going to compare the color change formed of test with that of the standard one. दोन्ही मध्ये प्रोसिजर टेस्ट सॅम्पल मध्ये as well as स्टँडर्ड सॅम्पल मध्ये प्रोसिजर सेम राहील. फक्त स्टार्टिंग मटेरियल दॅट इज टेस्ट सॅम्पल मध्ये गिवन सबस्टन्स असेल आणि स्टँडर्ड मध्ये ऍज पर प्रिस्क्राइब बाय फार्माकोपी असेल. बाकीचे इंग्रेडिएंट्स मात्र सेम राहतील. So in case of limit test for arsenic, the various reagents or ingredients used are potassium iodide, nascent hydrogen. So all this reagent, it should be free from arsenic. So arsenic present as an arsenic acid. Initially the arsenic impurity, it is present in the form of arsenic acid. In the sample, it is firstly reduced to arsenious acid. So firstly आपण substance मध्ये acid टाकणार आहोत. We are going to perform the limit test for arsenic in acidic condition. After addition of the acid, जे काही arsenic impurity present आहे substance मध्ये, that will get converted into the arsenic acid. So जे काही arsenic acid आहे, that will get react with the potassium iodide or it may either get react with the stannous acid or it may either react with the zinc or hydrochloric acid, etc. And this arsenious acid is then further reduced to the formed arsenious acid is then further reduced to arsine gas by reacting it with the nascent hydrogen. And this formed arsine gas will then react with the hydrogen of mercury chloride paper and it will give the yellow coloration or yellow stain. The depth of the yellow stain on mercury chloride paper will depend upon the quantity of arsenic which is present into the substance. So initially we are going to perform the limit test for arsenic into acidic condition. So impurity that is arsenic react with acid, formation of arsenic acid takes place. The formed arsenic acid will then either react with potassium iodide, stannous acid, zinc or hydrochloric acid and it will get converted into arsenious acid. This arsenious acid on further treatment with the nascent hydrogen. This nascent hydrogen is formed due to the reaction between these three, that is potassium iodide, stannous acid and zinc hydrochloride. So the formed arsenious acid will react with the nascent hydrogen, formation of arsine gas takes place. The formed arsine gas will then pass in between the tube, pinhole of the tube and it will get react with the mercury chloride filter paper which is placed in between the two rubber bunks and it will give the yellow stain. We are going from the depth of the yellow stain produced by the test sample with that of the standard, we are going to draw the results whether it passes the limit test for arsenic or not. So this is the basic principle behind limit test for arsenic. Firstly, we are going to convert the impurity into arsenic acid. Nextly, arsenic acid reacts with stannous acid, potassium iodide, zinc and hydrochloric acid. It will get converted into arsenious acid. Arsenious acid will then react with the nascent hydrogen, and conversion of the arsenic impurity, it will get converted into arsine gas. The arsine gas will then react with the mercury chloride filter paper, and it gives the yellow stain. So these are the steps by which limit test for arsenic is to be performed. That is conversion of arsenic into arsenic acid into arsenious acid when react with the nascent hydrogen formed in between the reagents into arsine gas. So this is the principle. Next, when the sample is dissolved in acid, initially the sample is dissolved with HCl. So the arsenic which is present in the sample gets converted into arsenic acid by the action of reducing agents, that is potassium iodide, stannous acid, and arsenic stannous acid, etc. The formed arsenic acid, it will get reduced to arsenious acid. The nascent hydrogen formed during the wetting period of 40 minutes further reduces the formed arsenious acid to arsenic gas, which reacts with the mercury chloride paper, giving a yellow stain. So this is the specific apparatus used for performing the limit test for arsenic. Initially, we are going to place the sample into this 120 ml capacity bottle. They have 120 ml. Since it is a comparison method, we are going to use two gadget apparatus, one for test, another for sample. First, we'll study the test for limit test for test sample. So initially, into an 120 ml wide mouth bottle, we are going to place the test sample which contains arsenic impurity. To it, we are going to add HCl. Nextly, we are going to add water. After addition of HCl, the arsenic, it will get converted into arsenic acid. And due to the nextly, we are going to after addition of HCl and water, we are going to add potassium iodide, stannous acid, and etc. After addition of the reagents, the arsenic acid, it will get converted into arsenious acid. This arsenious acid, हे सगळे जे काही रिएक्शन्स आहेत 3 स्टेपचे, all this get occurs into an 120 ml capacity bottle. So all these reactions occurs due to the presence of reagent, that is potassium reducing agent, that is potassium iodide, stannous chloride, HCl, zinc, etc. तर इनिशियली सॅम्पल असेल, HCl असेल, वॉटर असेल, नेक्स्टली पोटॅशियम आयोडाइड असेल, झिंक ग्रॅन्युल्स असतील, हे सगळे आपण 120 ml कॅपॅसिटी वाईड माउथ बॉटल मध्ये प्लेस करणार आहोत. वी आर गोइंग टू स्टर इट. नेक्स्टली जे काही बॉटल आहे, to that bottle we are going to attach an tube which is having 20 centimeter length or 200 mm of glass tube. मग ह्या ट्यूबला जे लोअर साइडला आहे त्या लोअर साइडला एक पिन होल असेल. व्हॉट इज द यूज ऑफ दिस पिन होल? जे काही अर्सेन गॅस तयार होणार आहे ड्यू टू द रिएक्शन ऑफ इम्प्युरिटीज, दॅट इज arsenic with that of the reagent, that arsine gas will get passed over this tube and it will get adhere on the mercury chloride paper. So this is the construction and working. So construction मध्ये 120 ml capacity bottle आहे. To it we are going to attach 20 centimeter glass tube. At the end of this glass tube, 2 mm diameter hole is present. So from this hole, the arsine gas formed will get passed to this tube. The internal diameter of this tube is 6.5 mm. In between of this tube, there is presence of the cotton plug. And this cotton plug is moistened with lead acetate solution. Lead acetate का use केलं? During the reaction of arsenic impurity with potassium iodide, zinc, or other reducing agent, there may be formation of impurity gas, that is H2S gas. जे काय H2S gas आहे, that will get adhere on this cotton plug moistened with lead acetate paper solution, and only the arsine gas will pass over this tube and it will get adhere on mercury chloride filter paper. So in between of the tube there is presence of an cotton plug which is moistened with lead acetate solution. At the end of the tube there is presence of 2 rubber bunks. One is attached over the another, and in between these 2 rubber bunks there is presence of filter paper which is dipped into the mercury chloride. So when arsine gas passes over this hole, from this tube it will get adhere on the mercury chloride paper. It will react with the hydrogen entity of the mercury chloride paper and formation of yellow stain takes place. Both these bunks are attached together with the help of spring clip. So this is the construction and working of limit test for arsenic. We are going to perform it into a special apparatus, that is gudges. Same procedure we are going to repeat it for the standard one. Only the difference will be the presence of the substance. Standard मध्ये limited amount of arsenic impurity आहे, which is tolerable one. Test मध्ये we don't know the presence of impurity amount. We are going to compare it with the standard one. If the intensity of the test sample produced is yellow stain coloration produced is less than that of the standard one, obviously it contains less amount of arsenic impurities and it is tolerable one and it is desirable one. And if the intensity of the yellow color produced by the test sample is more than that of the standard one, it contains more amount of allowable arsenic impurity and which is undesirable and harmful one. So this is the construction and working of limit test for arsenic. We are going to perform it into an 120 ml capacity bottle. Initially, we are going to take the sample. To it, we are going to add water, HCl, and other reducing agent, that is potassium iodide, stannous chloride, zinc, etc. And we are going to attach this 120 ml capacity bottle with a tube. This tube is having a hole at the bottom. हे जे काय tube आहे, it should not touch the surface of the liquid. It should be above the surface of the liquid. जर ते टच झालं तर liquid sample त्याच्यामध्ये जाईल and gas will not be able to pass through the tube. So this placement of this tube onto the 120 ml capacity bottle, we have to done it with a greater care so that its tip, it should not touch with the surface of the liquid. ह्या होल मधून we are going to wait it for a period of 40 minutes. During waiting period, जे काय arsenic impurity आहे, that will get converted into arsine gas. This arsine gas will pass from this 2 mm diameter hole in through this tube and it will get adhere on the mercury chloride filter paper, which is placed in between these two rubber bunks, and these two rubber bunks are attached with each other with the help of spring clip. Same procedure we are going to repeat it for the standard one. So this is the description of apparatus. Already we have covered it into the diagram, that is capacity of the bottle, diameter of the hole, length of the tube, internal diameter. In between of the tube we are going to place a cotton plug which is moistened with lead acetate. What is the use of lead acetate? It will adhere the formed H2S impurity. It will not allow the passage of H2S impurity from tube towards the mercury chloride paper. So next is the procedure for limit test for arsenic. So since it is a comparison method, we are going to perform the practical in two ways, one for test, another for standard one. First we'll study the procedure for test sample. So for a test sample, we are going to take an Gudget apparatus. So the test solution is prepared by dissolving specific amount in water and stanated HCl, that is arsenic free HCl, and we are going to kept into a wide mouth bottle which is having capacity of 120 ml. So initially that test Gudget sample मध्ये Gudget मध्ये sample असेल. For dissolving purpose, we are going to add water. For maintaining the conditions, we are acidic, we are going to add HCl. That HCl, it should be free from arsenic. Nextly to it, we are going to add reducing agent, that is 1 gram of potassium iodide. What is the use of reducing agent? It will convert the arsenic acid into arsenious acid. So 1 gram of potassium iodide. To it, we are going to add next reducing agent, that is 5 ml of stannous chloride solution. Lastly, we are going to add 10 gram of granulated zinc, and all the reagent used, that is potassium iodide, stannous chloride, and zinc, it should be free from arsenic and it should be represented by a letter, that is AST. After adding all the reagents into 120 ml capacity water, we are going to keep the solution for a waiting period of 40 minutes. During waiting period, the reaction gets occur. Arsenic impurity, it will get converted into arsine gas. This arsine gas will get passed over the tube. This arsine gas will get passed over the tube and it will react with the mercury chloride filter paper, that will give yellow stain or yellow coloration. Same procedure we are going to repeat it for the standard one. Only the difference is a known quantity of dilute arsenic solution. This known quantity is desirable one and that is not harmful one. We are going to dilute it with arsenic solution in water and we are going to add stennated hydrochloric acid. This stennated hydrochloric acid, it is an arsenic free and it is kept into a wide mouth and bottle. Nextly, we are going to add reducing agent, that is 1 gram of potassium iodide, 5 ml of stannous chloride acid, 10 gram of zinc, and all this reagent, it should be free from arsenic impurity. Nextly, we are going to keep the solution for wetting for a period of 40 minutes. During wetting period, this reaction gets occur. That is conversion of impurity into arsenic acid. The arsenic acid when then reacts with potassium iodide or stannous acid, that is H2SnO2. After reacting with this stannous acid, this arsenic acid, it will get converted into arsenious acid. This arsenious acid will then react with the nascent hydrogen. So how does nascent hydrogen is formed? Nascent hydrogen is formed due to the reaction of the potassium iodide and zinc. So formed arsenious acid reacts with the nascent hydrogen, formation of arsine gas takes place. The formed arsine gas will then pass through the hole that is 2 mm diameter through the tube. It will then react with the mercury chloride paper and will give the yellow stain or coloration. So stain obtained on mercury chloride paper is compared with the standard solution, that is with that of the second Gutzet stain. And standard stain, it should be freshly prepared as it fades on keeping. म्हणजे जे काय स्टेन तयार होणार आहे mercury chloride paper वर ते फेड होऊ शकते. So both the test as well as standard solution is to be performed freshly and simultaneously. We are going to compare the yellow stain produced by the test with that of the standard one. Inference is if the stain produced by the test is not deeper, म्हणजे fade असेल किंवा कमी असेल than the standard stain, then the sample complies or passes with the limit test for arsenic. Jर stain हे test चा कमी असेल as compared to standard one, so it passes the limit test for arsenic. So this is the in detail basic introduction, principle, construction and procedure of limit test for arsenic.